Thank you. Hello, everyone. Thank you for joining us this afternoon. For those of you who I don't know yet, my name is Lata Palniban, Professor of Medicine in Primary Care Population Health. As we begin today, I would like for us to take a moment to acknowledge the land on which we sit here at Stanford Medicine.
I will share my screen, Martha.
Stanford sits on the territory of Huichin, the ancestral and unceded lands of the Muwekma Ohlone tribe,
who are the successors of the historic and sovereign Verona Band of Alameda County. This land was, and continues to be, of great importance to Native people.
We recognize that every member of the community has benefited
and continues to benefit from the use and occupation of this land.
Consistent with our values of community and diversity,
we have a responsibility
to
acknowledge and make visible
the university's relationship
to Native people.
Thank you. In an effort to build support and community among Stanford community members who are engaged in health equity, Stanford Medicine's Office of Faculty Development and Diversity has launched the Health Equity Action Leadership Network, known as HEAL, along with a series of events throughout the academic year. As a member of the HEAL Network Steering Committee on behalf of OFDD, I would like to welcome you all to this HEAL Network event, Health Equity Research in Genetics. The HEAL Network aspires to provide health equity research collaboration opportunities and foster a community and a network among Stanford affiliates engaged in health equity, offer education about numerous health equity topics, unveil health inequity and injustice, and equip Stanford community members with tools to transform health systems. Today's event will feature 3 fireside chats, followed by an open Q&A. Please type your questions into the Q&A feature even as we're going through the talks. We will be recording the fireside chat, but will stop recording once Q&A begins. Feel free to submit questions via the Q&A feature, and we will answer those questions after the fireside chats. We have 3 faculty who will share their experiences with health equity research in genetics. I will now take a few moments to introduce our first presenter, and I will introduce each of the presenters before their session. It is my honor to first introduce Dr. Mildred Cho, who is a professor of pediatrics and the principal investigator of the Center for Integration of Research on Genetics and Ethics, known as SURGE at Stanford University, which is funded by the NHGRI of the NIH. She is the Associate Director of the Stanford Center for Biomedical Ethics. Her major areas of interest are the ethical and social issues raised by new technologies such as genetic testing, gene therapy, pharmacogenetics, and gene patents. She also studies how academic industry ties affect the conduct of biomedical research. Thank you, Dr. Cho, for your presentation today.
Thank you. So thanks for having me at this fireside chat. I'm going to talk a little bit about the use of race and ethnicity as categories in genetic research. And doing this, I'm going to be informed about my perspectives recently as a member of a committee of editorial board members for the journal Genetics in Medicine that was charged with developing policies and procedures for the journal to address equity, inclusion, and diversity issues. but my views represent my own and do not represent those of the journal. Using race and ethnicity as categories in research is necessary and important to studying health equity and disparities. But in practice, that's not typically why these categories are used. They're often used as biological markers of disease. But the use of socially defined variables such as race and ethnicity in this way has been rightly criticized for decades, yet the practice persists. In this article that I'm showing here on the screen, this author, Jay Cohn, defends the use of biological variables in this way, calling on how race and ethnicity and racial distinctions in clinical research reveals differences in mechanisms of disease and disease frequency, and the role of those categories in diagnostics and therapeutics. And in the article, the author defended the practice by saying that we should not throw the baby out with the bathwater. So in response, I wrote this article, and this was a long time ago. This was in about 2006, and responded with my own argument that there is no baby in the bathwater. So even if clinically significant differences are associated with race and ethnicity, it's important to remember that association is not causation, especially if important variables such as socioeconomic status are not included in analysis. So while some clinically significant differences may map onto race or some race or ethnic boundaries, many also do not. So there's many reasons why these categories of race and ethnicity are problematic in genetic research and biomedical research in general, not least that they are very imprecise and non-scientific. Furthermore, if they're not used to measure socially determined phenomena such as racism, the focus on biological correlates of race detract from our ability to identify sources and determinants of inequity. So I want to acknowledge my co-authors on a paper that we wrote recently, who these co-authors, Kyle Brothers and Robin Bennett, we are all editors for the journal Genetics in Medicine. And we wrote this article as part of our efforts to launch our anti-racist journal policy development, as well as wanting to point to some other work that informs what it means to promote health equity in genetic research through the reporting and also study design of that research. And I also want to point to this article that some of you may have seen that was published in Health Affairs in the blog by Rhea Boyd and colleagues that inspired us at Genetics in Medicine to write this article on taking an anti-racist posture in genetics and genomics research. So I just want to point to a couple of examples that show what's at stake in the accurate use of racial and ethnic variables in genetic research. So one is that we tend to over-attribute importance to race that could lead to problems with diagnostics or treatment decisions. So these are a couple of quotes from an article that talks about pharmacogenomics, and it says how 5 to 10% of Europeans, but only about 1% of Japanese have a loss of function variant at the CYP2D6 locus, which is a genetic locus that affects the metabolism of many drugs. So that implies that there's a sort of ethnic difference or a racial difference or an ancestry difference here that is significant in terms of using these drugs. But later in the same article, it points out that the alleles at the CYP2D6 locus also vary in frequency even within Europe, from about 10% in northern Spain to 1 to 2% in Sweden. So that basically undercuts their sort of implication in the first part of the article that race or ethnicity was an important marker of that's important to know about in terms of pharmacogenomics. Another example that many of you may be familiar with is that race or ethnicity has been used to, for example, in guiding testing decisions or in guiding diagnostics for sickle cell disease, with the idea that, at least in the United States, it tends to be found more frequently in people of African descent. But this is potentially misleading because some of the highest rates of sickle cell disease are actually not in Africa. In fact, in India, parts of Southern Europe, and Central Europe. And on the African continent, the prevalence of sickle cell trait in specific areas has a very large range, from less than 1% to 45%. So the use of race or ethnicity can be very misleading in a case like this. In addition, the use of racial and ethnic classification is very inconsistent by people, even in describing themselves. So, although I think sometimes we have the temptation to say, well, why don't we just let people define themselves in terms of what their race and ethnicity is? That's not necessarily helpful. So, in this article that was published about 20 years ago, actually, that used 1990 US Census data on those indicating that they were of Indian ancestry or birthplace in India, found that 10% of them actually identified themselves as white, Black, or American Indian, despite instructions directing them to the Asian Indian category that was supplied at the time. And speaking English among that group was associated with higher likelihood of self-identifying as white or Black, as opposed to Asian. Interestingly, those of Indian national origin were more likely to select Black, while those of Sri Lankan or Pakistani origin were more likely to select white. 80% of the children with one white or one Asian Indian parent were listed as white. So you can see that even across generations within the same family, race and ethnicity is not consistent. So what can researchers do about this? And one of the things that we're asking researchers and authors to do in submitting their manuscripts to Genetics in Medicine is to justify the use of race or ethnicity as a variable in a way to encourage people to really think about it rather than just do it as a sort of knee-jerk reaction, and to be rigorous about using these variables in terms of defining it, describing how it's ascertained, paying attention to validity and reproducibility issues, just as you would with any other scientific variable. And finally, to critically examine how race or ethnicity is used in studies, and whether it's being used or implied to be a biological variable, which is inappropriate, or the cause or marker of a disease just because it's got an association. And to think about whether there are other important variables, such as social determinants of health, that were not studied that perhaps should be studied in terms of making the conclusions that you want to make. So I'll stop there with my slides so that we can move on to the next.
Thank you, Dr. Cho.
Presentation.
Thank you, Dr. Cho. So our next presenter will be Dr. Clark, who's a preventive cardiologist and an instructor at the Stanford University School of Medicine in the departments of medicine and pediatrics. Completed clinical training in internal medicine at Brigham and Women's Hospital, pediatrics at Boston Children's Hospital, and cardiovascular medicine at Stanford Hospital. He is board certified in all of these three specialties. His research is focused on understanding complex disease genetics in diverse populations, integrating monogenic and polygenic risk with clinical risk, and large-scale phenotyping using the electronic health records. His clinical practice focuses on identifying risk factors for cardiovascular disease with the goal of promoting health and longevity through evidence-based, personalized treatment. He is interested in developing family-centric approaches for the treatment of adults and children carrying high genetic disease risk. Over to you, Dr. Clark.
Okay, thank you. Thanks for having me. And thank you to Dr. Cho for that great talk, which really serves as a nice segue into my talk, because I will show some analyses that look at race and ethnicity, but the point of those analyses are specifically to understand disparities driven by structural racism in genomic medicine. So I have no disclosures, and the three main things I'll address today at the request of the panel was my general research focus, a few specific projects, and then I'll touch on my personal journey, which I've gotten to hear other HEAL Network panel speakers discuss, and I've found it to be very inspiring, so I thought I would share mine as well. To start off, I think most of us know that coronary artery disease is the leading cause of death in the United States and globally, and in both men and women. But we sometimes think or mistakenly believe that it's a disease of the old, when in fact, coronary artery disease begins affecting people very early in life. In the early 30s, it's the 5th leading cause of death. By the early 40s, it's the 3rd leading cause of death. And by the early 50s, it's the #1 cause of death in the United States, and it remains that way then on out. But preventing these early deaths from coronary artery disease is challenging because many of the approaches we use to prevent coronary disease and its complications are focused on older populations. And that's one of the reasons why people have gotten very excited about the idea of using precision medicine and genetics and genomics to better identify very high-risk individuals earlier in life, so that we can work to prevent those deaths. And there's been articles in the lay press as well as in the scientific press about using genetic risk scores or polygenic risk scores for this purpose. For today's brief talk, I'm not going to go into how polygenic risk scores or polygenic scores are developed. I'll leave it as sort of these black boxes, but the key steps is first, a genome-wide association study must be performed to understand genetic associations. These are often not the causal genetic variants, but markers of risk in a population. Another population is typically used then to train a polygenic score, and then a third population would potentially be used for validation. And we've gotten to the point over the last 10 years where the interest in polygenic scores have rapidly increased. And in fact, we've now had studies of looking at real clinical applications of these scores, and there are some clinical trials that are underway of using polygenic scores for coronary artery disease, for example. There will be some pilot studies. In fact, at Stanford, there will be some pilot studies rolling out in the next year using polygenic scores in the context of clinical medicine. But there's an issue at hand, and that's that the data used to develop these polygenic scores is incredibly Eurocentric. And if we look at, in blue, the genome-wide association studies that have gone into developing the available polygenic scores, so this is counting the number of scores that have used data from a given genome-wide association study, and on the right is the number of participants. You can see that the studies derived from European populations dwarf others, and particularly when it comes to the step of training and validation, there really has been no studies to date, particularly for coronary artery disease, to appropriately train and validate the use of polygenic scores outside of primarily European ancestry populations. Now, what is the impact of that? And this is an example of what I meant in terms of using, in this case, self-identified race ethnicity to compare how these scores work in one group versus another. These are US adults. The data comes from the Million Veteran Program. And we're looking at individuals. Here is the highest score, the most at-risk individual by the method of a polygenic score versus the first quintile would be the lowest score. And the non-Hispanic whites, we see a spread of risk and the score's ability to predict risk per one standard deviation and score. There's a hazard ratio of 1.37. This is the current best score for for coronary disease prediction. When applied to non-Hispanic Blacks, we don't get that same risk stratification. And this isn't because there's any biologic difference in the pathophysiology of coronary disease in non-Hispanic whites and Blacks. It's the same pathology. And even the genetics is essentially the same, but the markers of risk that get used and sort of prioritized in the process of developing a score do not work universally, because each of us, our genetics will reflect the historic migration of humans in our past, our ancestors, where they lived and where they moved. And those patterns of migration affect certain underlying features of the genome related to correlations between variants. And so without adequate representation, these scores don't work well when taken from one population and applied to another. This is part of well over a decade now of very clear and striking disparities in genetics research in terms of representation. And it has started initially with genome-wide association studies. It's progressed into development of polygenic scores, and it's now being applied clinically through the application of these scores. And at each step, there's a sort of self-amplifying cycle where the existing structural racism in the methods for developing new scores, it sort of feeds back on itself and it propagates forward. So here are 3 projects that I'm working on to try to address some of these issues. One is to simply improve polygenic scores specifically for the outcome of coronary disease. And this is by using more diverse genome-wide association studies and then leveraging those studies to construct scores that hopefully can be used more universally. This is very preliminary work. I haven't actually advanced it yet to some of the more sophisticated techniques, but this is sort of a first pass to give a sense of what can be done once you introduce some level of diversity into the GWAS. There's a lot of work still to be done for improving how these scores function, at least for coronary disease and non-Hispanic Blacks, but we can actually see a great improvement for Hispanics. Another method for improving risk prediction is to use genome-wide association studies for coronary disease risk factors. This is for LDL cholesterol as a risk factor. And what we can see, looking at an older score that was derived from a European GWAS, it performed very poorly when applied to non-Hispanic Blacks and not as well in non-Hispanics compared to whites. A newer score we've developed using a multi-ancestry genome-wide association study and a multi-ancestry meta-analysis has helped to improve the performance across all 3 race ethnicity groups. And then lastly, rare variant analyses. There is no such thing truly as, in my opinion, a population-specific genetic variant, because any person who carries a genetic variant, if they pass it on to their offspring, it can basically be part of any population. Populations are not discrete structures. That being said, rare variants may be more frequent in one group than another, especially when there's, for example, founder populations. The classic example is familial hypercholesterolemia, and there's certain founder populations in, for example, Canada, French Canadians who have a high frequency of FH variants and other populations with lower frequencies. So I've been harmonizing data across many NHLBI studies that are quite diverse compared to prior research in order to better identify rare variants across many cohorts in the United States. So with that, I'll then just say a few words about my own journey here. Like many people, my story starts with my mom. This is my mom, Diane Clark, her father, William Clark, and her mom, Georgia Wells. She was born in in Boston and raised in the Jamaica Plains projects. I never met my grandfather who passed away before I was born, but my mom unfortunately did not have a great childhood and spent much of her life bouncing between homelessness and foster care. She then eventually escaped Boston to Portland, Maine, which is where I was born, and she raised me and my sisters. And having left school at the age of 13, she did not have a lot of educational resources, and as you can imagine, that did not make it easy. And we, as a family ourselves, spent time in living in homeless shelters and so forth. But thanks to many courageous efforts by my mom, I was able to then go to Cornell University after I finished high school. At Cornell, I met this man, Carlos Bustamante, and he gave me this book called The Mismeasure of Man by Stephen Jay Gould. And it was through reading this book and my discussion with Carlos that I got interested in genetics. I had gone to Cornell with the plan to go into medicine. I had no understanding of genetics or research or even what a PhD was. But he got me both excited about genetics, but also, like him, very concerned about how genetics could be misused in medicine or otherwise to propagate racism and racial disparities. So thanks to his mentorship, I graduated from Cornell. Here I am with my mom and my aunt and my cousin. I came out to Stanford, and at Stanford, I completed the MSTP program. I got my PhD in genetics. I graduated. Here I am with my sisters and my mom. And after completing med school, I went on, as we heard at the beginning, to do residency training in Boston. And then I'm now back here as a cardiologist and as a genetic researcher. And if anyone is interested in any of the projects I mentioned, please reach out to me. My email is very easy. It's just my name, Shoa, S-H-O-A, @stanford.edu. So I'd just like to acknowledge my key collaborators in the work I showed, as well as the consortia I'm a part of in the funding I recently was able to get through the Diversity Chair Investigator Award. So thank you.
Thank you, Dr. Clark, for your inspiring comments and story, and for sharing your story with all of us. And thank you for sharing that wonderful cutting edge work in genetic diversity. And I'm sure we'll have lots of questions in the Q&A about your work. So thank you. And now it is my honor to introduce Mr. Joseph Iracheta, who is an Amerindigenous scientist from the Purepecha and Rarámuri from Mexico at the Native Biodata Consortium, NBDC, with the Cheyenne River Lakota Nation, a Sioux tribe. Mr. Iracheta has been a scientist since 1990, where he started as a bench biotechnician and continued in many science disciplines. In 2014, he graduated from the UW Seattle with a Master's of Pharmaceutics and Bioethics under Drs. Ken Thummel and Wiley Burke. He is currently finishing a DRPH in Environmental Health and Engineering from Johns Hopkins under Drs. Anna Navas-Asian and Paul Locke. Mr. Iracheta is passionately working to end Amerindigenous health disparity by wearing many research hats of law, ethics, policy, genomics, health outcomes, epidemiology, healthcare prevention and intervention, and allostatic load from systemic racism. Mr. Iracheta believes that all data and resources must be seen as unforeseen futures where their value will constantly change. He feels this data must be secured for indigenous economic sustainability. Thank you, Dr. Iracheta, for joining us today, and I'll turn it over to you.
Well, thank you for making me a doctor before March.
Almost there.
Yeah. So my last name is Iracheta. It's a Basque name from Spain, but I don't have any Basque ancestry. I'm a Mexican indigenous person from the Altiplano, which is Michoacan, Jalisco, Guanajuato and Querétaro in Mexico. And my wife and kids are enrolled on the Cheyenne River Sioux Reservation. You'll see at the bottom of this slide my links to Twitter. And the title of my talk is Reclaiming Genomics and Data Science for Indigenous Sovereignty. So in reference to the previous two speakers, there's a lot of things that are of great promise with genomics and precision medicine, but there's a lot of peril as well, specifically capitalism, intellectual property, and the theft of native resources. So that's basically what my talk is going to be about, in addition to the inaccuracy of precision medicine, as Dr. Shoa just showed us, in terms of the polygenic risk scores. So this is just a close-up picture of me. I do talks locally on native radio stations, and we have done a couple of videos and radio shows. We were on Science Friday, we are on the CBC Ideas, and then we have a website where we've done some videos, and we're slated to be in upcoming documentaries as well. I went to Loyola Chicago. I worked at UW-Madison. I went and got my pharmaceutics degree at UW-Seattle, and I'm currently finishing at Johns Hopkins. I'm the executive director and vice president of the Native Biodata Consortium in South Dakota. Cheyenne River Sioux Reservation is north central South Dakota in Eagle Butte. And I am from Chicago, and then Hopkins is on there. The rest of these are my partners and where they're located in the United States. So why is the NVDC necessary? Cultural sustainability, economy, health for both the people in the land, and sovereignty. So we all know the horrible depredations of the American government on Native American people, but what people don't know is that that is continuing today. And the strongest example is the pipeline fight for Standing Rock in North Dakota. That was 8 months long with 15,000 people. And it was not only hampered by a lot of fake news by the local sheriff's department and the state militia, but the signals and all kinds of other things to get the real news out was blocked by the US Army. So these things are still happening. It's just not talked about. So in terms of dissolution of cultural cohesion, there's lots of different ways that's happening, and it's fractionating the Native people to exhaustion. So in terms of both Dr. Clark and Dr. Cho's presentations on race and ethnicity, Native Americans are a broad group, including a lot of Latinos and especially Mexican Americans. So Native American isn't one solid umbrella, and they have different amounts of admixture. They have pre and post-Columbian differences. There's all kinds of reasons not to lump them under one umbrella, which makes precision medicine very difficult. But in terms of politics, there's a lot of talk about urban natives versus homeland natives. And currently that's what's happening with the All of Us program, is limiting the jurisdiction of native control over their own DNA only to their tribal borders and not anywhere else. Also American citizen versus tribal citizen, they pit their own citizens against their tribal governments. American Indian versus indigenous of the Americas, because as I said, from Alaska to the tip of South America, we share a lot of the same genome because our genomes are special and unique evolutionarily over the rest of the world's population. And then of course, with the common rule, individual rights and consent are always at odds with tribal sovereignty and approval. This is just a short slide to show all the different ways that distrust happens, differences in values, cultural and spiritual concerns, lack of interest in research, both from both sides of the kind of research that needs to be done, and then political as we saw with the Trump administration, he was terminating the tribes he didn't like and then giving recognition to the tribes that he did like. Unforeseen effects from DNA on federally recognized rights, stigmatization, and racial profiling. They're small, easily identified by zip code and by long runs of homozygosity. And then there's the ever-present fear of biopiracy and biocolonialism. This is just one example of all the different locally adaptive traits from different small populations around the world that will mean billions of dollars in terms of pharmaceutics, and Indigenous people will not be able to take part in any of that profit. Data is the next big trouble issue. We know that in addition to publicly funded research, Google, Facebook, and Microsoft all have their own biomedical genomics programs, and the digital genome market is going to skyrocket here in the next 6 to 10 years. Also, Native Americans suffer the worst health disparities in the country. This is their own homeland. These are their own foods. These are their own lands, and they suffer the worst health care from IHS and the worst health disparity from lots of other socially determined variables. And then we always have this yo-yo between the different administrations, between making sure that tribes assimilate versus giving tribes the right to be self-governing. And in both the Clinton and the Obama administrations, they supported treaty and constitutional law by sending out executive orders. This was the last one that was done by the Obama administration to make sure that anything that was not specified in former documents belongs to the tribe. And because it belongs to the tribe, you have to consult with the tribes before doing anything that might harm them culturally, their land-based, economically, politically, and that has not been done. But here Biden is promising to reassert that commitment to tribal sovereignty. So the question should never be, how do we get tribes comfortable with genetic and other data? The question should be, how do we protect tribes if they want to share their data for their own benefit? So lately we've had all these conversations about inclusion and diversity, but that does not mean equity, because including people in studies doesn't make sure that they're going to get the interventions that they need on the other side, because the other side is all based on money. So this is what we're usually told, is that phase one, we're going to collect some wonderful data to help you for community benefit. But the second part isn't really worked out, and the federal government doesn't care or hasn't tried to work that out. So what actually has happened in the past and is going to happen in the future is that there will be not community benefit, there'll only be profit to somebody else. And this includes our DNA and other ancillary data as big data grows. What happens is, what has been happening is that even though tribes have long been the only ones with government guaranteed healthcare, they do not get good healthcare. They haven't in the past and they don't now. They get half the money spent on them per person than other people in the United States. And what happens in research often is they get a altruistic reason to participate in research, told they're going to miss out on benefits. And if they don't engage, they're going to increase the power imbalances. But instead, some of them participate, then they create research harms, and then it causes Indigenous people to disengage from all kinds of research. And the cycle just keeps continuing and continuing and continuing. There's all kinds of crises in Native American communities, including education, so that we can't do this for ourselves. The assumption is that we're incompetent and that we can't do these things for ourselves, especially during COVID. And a lot of things since 1998 and 2008, when there were two world market crashes, have caused people to go back into what's left of Indigenous spaces around the world to ramp up capitalism over again. So it's the second era of discovery. So basically, we are paying for your party. So how did the NBDC start? It started with a lot of young, wonderful Indigenous scientists throughout the country, in New Zealand and Australia and Canada, which we all convene at the Summer Internship for Indigenous Peoples and Genomics, which started in 2011. And then we started locally with an autoimmunity study for the Lakota. So these are my partners, Katrina Claw, she's now at the University of Colorado, and Keolu Fox, who's at University of San Diego. She's Navajo and he's Hawaiian. We were members of the UW SACNAS, which was a unique group because they were all graduate students and no undergraduate students. And we've won all kinds of awards and we did all kinds of public community service. But there wasn't a journal club for just Indigenous people. So we started one, which was just us three. And we conceived of the Native Biodata Consortium back then. And we are glad to have it come to fruition. Singh was also instrumental. Singh has been graduating more PhDs in public health, genetics, MDs, ethics, law, because they're empowered to take up data for tribes. It's Indigenous data sovereignty, that's the movement. And we've spread through Canada, Australia, New Zealand, and soon to be Mexico. As I said, we started with these partners, starting the study of autoimmune illness in the Lakota. Those of you that don't know, the autoimmune diseases that are studied the most are European, specifically Northern European autoimmune diseases, but autoimmune diseases in Native Americans are relatively unstudied or understudied. So the NBDC, who are we and what we are? This is a picture of our lab. Actually, we have more equipment now. Our goal is to create data sovereignty, data utility, and data solutions locally by leveraging the interest in Native American data by keeping the data local. So we want to, even though we want to do precision health and omics, we want to really emphasize the other two bubbles, which is data sovereignty and policy and sustainability, and real community engagement, because a lot of community engagement that's going on right now is just a checked box and it's not real community engagement. Real ethics, or making ethics tailored to Native communities, because the Common Rule does not serve Native communities, advancing tribal regulation and education and capacity building. This is the outside of our building. We're the first Indigenous-led biological and data repository within tribal jurisdiction. We're a 501c3. Our board members are almost all Indigenous, and our community advisory group is only from Cheyenne River. We have many research partnerships, and we are trying to harness the power of tribal jurisdiction and authority to prevent the incorrect arguments that we're all in this together. We got donated a lot of equipment by Illumina. We have an XSeq sequencer. We have a 3-year maintenance contract. We have a 3-year guarantee of reagents to do not only COVID testing, but also sewer water capture for COVID and soil microbiome and agricultural testing. So here's a picture of the NextSeq. Here's a picture of Illumina installing everything for us. Like I said, we're doing COVID. We are including not only community engagement and policy to a CLIA certified lab, but we're also doing health education capacity and workforce development, and then sustainable health resources by collecting and keeping that data there for future projects. So we want to invalidate the presumed incompetence in our communities and dismantle the just sit tight until we get to you, which is what happened with COVID. We had to wait until August to get any tests. And by that time, South Dakota was overrun with COVID cases. So we are going for CLIA certification so that we don't ever have to wait for the CDC again, that we'll do it ourselves, and we'll take our partners' new technology under EUA and get them out into the community. We're also teaching. We did our first Indigidata last summer virtually. This summer we're going to do it in person, where we talk about all kinds of data from beekeeping to GIS or GPS data, all the way down to the microbial level. So all data and how it can be protected for the Indigenous space. These are some of the papers that
you'll find 8 publications that we.
Oops, sorry about that. These are some of the papers that we've done so far, and I wanted to call your attention to
the publications.
Call your attention to number 5 specifically, because the polygenic risk scores are likely not to work for Mexican populations. So just like Native Americans, Latinos are not one body. There's several different ethnicities, And so you can't lump all Latinos into one box. So our global message is we must reconcile the widening disparity between practicing science upon Indigenous people and transforming science with Indigenous people. We can no longer be passive participants in science. We wish to elevate attention to Indigenous data sovereignties that is commensurate with science's exploitation of Indigenous genomes. Indigenous peoples have sovereignty over their data. Funding agencies and universities must stop feigning ignorance over this fact. And this is a paper that we wrote in 2021, February. And I thank you for your attention.
Thank you, Mr. Iracheta, soon to be Dr. Iracheta, for those enlightening statistics and really highlighting the disparities in Indigenous communities and the research that is being done with Indigenous communities. And thank you for your leadership in this arena. So now we have just about 10 minutes for a few questions, and I will have the first question for Dr. Cho. So Dr. Cho, thank you so much for bringing to light sort of the classifications of